Okay, so in this video, we're going to look at all of the essential knowledge that is listed in the curriculum framework for AP Bio. So this is Unit 2, Cell Structure and Function, topics 2.1 all the way to 2.11. So let's go ahead and start, So organelles and subcellular structures and the interactions among them support cellular function. So when we look at, I believe that's probably like the enduring understanding, the big picture in this topic, 2.1. So if we look at ribosomes, ribosomes comprise or are made of ribosomal RNA and protein. Ribosomes synthesize protein according to the mRNA sequence. So we can see that over here in the GIF on the left, where like here is your RNA sequence. And so depending on that RNA, what the directions say will determine which amino acid is brought. So basically, ribosomes make proteins based on the mRNA. We have two kinds of ribosomes. You have your bound ribosomes that are attached to the ref and you have ribosomes that are freely floating in the cytoplasm. So ribosomes are found in all cells. It is something that reflects common ancestry, so we find ribosomes in prokaryotes as well as eukaryotes. However, those ribosomes in prokaryotes are like a slightly smaller size, but they function the same. Now, the cool thing I want to point out about ribosomes as well is that you find them in mitochondria and chloroplasts have their own ribosomes, and that's because mitochondria and chloroplasts evolved from ancient prokaryotes millions of years ago. And when we look at ribosomes, that connects us to our rough ER and a conversation about rough and smooth ER. But here on the rough ER, you can see that there are ribosomes located there. So the ribosomes you find on the rough ER are part of the endomembrane system, where any proteins that will become part of the cell membrane or be exported, am I saying that right? Sent out of the cell by exocytosis will be made on those bound ribosomes. You might have free-floating ribosomes, but there are free-floating ribosomes as well that will you can find in the cytoplasm, and those proteins made, so like right here, those proteins made by those ribosomes will stay within the cell. Okay, now for the ER, we have two kinds. We have the rough ER and we have the smooth ER. And basically, the rough ER compartmentalizes the cell. So it is, you have here in the rough ER, this ER like lumen. Lumen is the general word for like space, I guess. But basically you have these ribosomes that are going to make proteins, and they can travel through that ER lumen. And now you have the smooth ER. This area that is missing ribosomes attached is the smooth ER. And the smooth ER synthesizes lipids, but it also detoxifies our cells. So if you take in like alcohol or something or other kind of toxin, it'll be enzymes located within the smooth ER that will detoxify your body. Now, kind of cool that if you really think deeply, it's going to be the rough ER and the smooth ER that work together to make the building blocks of our cell membranes. So the endoplasmic reticulum provides mechanical support and carries out protein synthesis on membrane-bound ribosomes and plays a role in intracellular transport. Oh, I love lysosomes. So here, lysosomes are membrane-enclosed sacs that contain hydrolytic enzymes. And if you remember from Unit 1, hydrolytic means like hydrolysis or breaking apart. So here you have some lysosomes. Now, if we look at this picture, you have your rough ER, and there's proteins made on the ribosomes there, and little vesicles will carry those proteins to the Golgi. Now, those proteins could be hydrolytic enzymes. Now, they'll be modified and like finalized in their structure within the Golgi. And then if you see here, like little vesicles will bud off of the Golgi. And because the Golgi is also membrane bound, meaning it's surrounded in a lipid bilayer. That lipid bilayer around that vesicle is now has those enzymes that are hydrolytic trapped on the inside. And the cool thing about lysosomes is that they are actually made from budding off of the Golgi, and the inside of a lysosome has a pH of 5. Now this is significant because out in the cytoplasm, the pH is 7. So if any of those hydrolytic enzymes were to kind of leak out of that lysosome, then your other organelles would still be safe. Because you need to remember, lysosomes are hydrolytic, and they will help to break down materials within them. So lysosomes are important in intracellular digestion. So this could be true for single-celled organisms like an amoeba or something that engulf. So this, you can see this food particle is being taken in by endocytosis, and now it's in a food vesicle. This right here is made of a lipid bilayer, and that lipid bilayer will fuse with the lipid bilayer of a vacuole, of a lysosome. So here you can see how the two, pretend here's the food vacuole and here's the lysosome that's also membrane bound. When they come in contact, their lipid bilayers will join, and now the inside of the lysosome is in contact with the inside of the food vacuole. And so now those hydrolytic enzymes can digest and break down that food. Hold on, let me hit pause and tell my kids to stop talking. And then you can see that this digestive vacuole now can then move to that cell membrane, and then by exocytosis send out those particles. Now, lysosomes can also be used, if you have a damaged mitochondria, for example, it can take that in, destroy it, and then recycle the macromolecules from that mitochondria. So basically, it's breaking down by doing hydrolysis and breaking those bonds. It's also involved, in apoptosis or programmed cell death. Then we have vacuoles. Vacuoles are also membrane bound, meaning they're surrounded in a lipid bilayer that have many roles. In plants, they serve multiple functions. Now, we always think about them as just storing water, but the vacuoles can also store macromolecules as well as waste products. And so that vacuole is not just like a water tank. It can also store ions. And in plants that undergo a specialized form of photosynthesis, like C4 plant, I think it's CAM plants. CAM plants can use that vacuole to store organic acids that they can use for the Calvin cycle. They've evolved a specialized way. So anyway, the vacuoles are important for storage and releasing of macromolecules. Now in plants, it can take up to 50% of that cytoplasm could be a vacuole, and that is important in turgor pressure, which basically is pressing on the cytoplasm of the plant, helping it to stand up straight. Now in animal cells, we don't have that large of a vacuole, but we do still have vacuoles. Ooh, chloroplasts. We have whole videos in Unit 3 on the chloroplast. And so chloroplasts are specialized organelles that are found in algae and plants, or photosynthetic algae, and they have a double membrane, and inside they have thylakoids where photosynthesis occurs, or the light reaction, and those thylakoids are surrounded by a stroma. Now, the Golgi is also membrane bound, surrounded in a lipid bilayer, and it's part of our endomembrane system. And that endomembrane system is basically the path that a new protein takes. So when you have a protein made on a ribosome on the ref-ER, little vesicles that bud off will carry that protein to the Golgi. And within the Golgi, they'll be packaged, modified, sorted, and then they can be carried in a vesicle to the cell membrane where they can be sent out by exocytosis. Now, sometimes let's pretend that the ref-ER, though, has a protein that it requires. well, the Golgi can actually send the protein back to the Refiar. Okay. Mitochondria have a double membrane, and they also have, when we talk about the compartments within a mitochondria, so these folds are the cristae, and the cristae, you can see here, basically increase the surface area for lots of cellular respiration to occur. Or aerobic respiration, the electron transport chain. We'll talk about that more in Unit 3. And when we look at the mitochondria, though, in this space here, the intermembrane space, that's where the proton gradient accumulates. Now, it should also be mentioned that mitochondria have their own ribosomes, they have their own plasmids, and they make their own proteins. And so prokaryotes, they did evolve from mitochondria evolved from prokaryotes millions of years ago. And yep, okay. Now that folding of the inner membrane increases the surface area to allow more ATP to be made. I like this picture because it just shows, it's just pretty. And then here's those plasmids, and here you can see all the ATP being made from ATP synthase on that inner membrane. So now hopping back to the chloroplasts. The chloroplasts, within them, they have the thylakoids. These are the thylakoids, and around the thylakoids, you have the stroma. And that is where the light and dark reactions happen in photosynthesis. So here is the thylakoid membrane. You can see little photons of light here in this GIF. And then in the outside spot, you have the stroma. Membranes in the chloroplast or in the thylakoid, they do contain pigments. So here are our photosystems in that thylakoid membrane. This will be discussed more in Unit 3. And then the Calvin-Benson cycle happens in the stroma, which is the space outside of those thylakoids. Now in our mitochondria, we have our matrix, and the matrix is where the citric acid or the Krebs cycle happens. So the double membrane of the mitochondria, you have your inner membrane here with the folds and the cristae, and then here you have the outer folds. So these can create two different like compartments within this organelle to help them become like specialized. So you have your Krebs cycle in one area, and then you have your proton gradient in another. And so establishing that proton gradient can help lead to ATP synthesis. So in this picture here, you can see that proton gradient and the electron transport chain located within that inner mitochondrial membrane, and how that Krebs cycle is happening within the matrix. Surface area to volume ratios affect the ability of biological system to obtain necessary resources, eliminate waste products, acquire or dissipate thermal energy. That's a fun topic. And otherwise exchange chemicals and energy with their environment. Now, one of my favorite little things is when you germinate lettuce seeds or radish seeds. So if you go to Home Depot or you go somewhere that sells seeds, I recommend you getting a little packet of lettuce and just put them on some wet paper towel for a few days, and you'll see the little roots start to germinate. And oh my gosh, all these little hairs that show up, they're so beautiful. Now, at first you might think it's fungus growing on your new roots, but in reality, those are like extensions of the root called root hairs. And their job is to increase the surface area for obtaining nutrients and eliminating waste for that germinating root. Now, if you think about what is happening when a root is germinating, oh my gosh, mitosis is happening like crazy to build all those new cells. Think about how metabolically, what is the word? Metabolically active? I don't think I'm saying that right, but like think about like how much activity, there you go, is happening within each of those cells as they're rapidly dividing. Think about all the ATP that would be required to build all the building blocks, right? So with that, if the plants are absorbing water and oxygen through their roots, like think about like the mitochondria requires oxygen for aerobic respiration. That has to diffuse in. And so with that, these root hairs are increasing the surface area, making that diffusion of oxygen more efficient. So anyway, I love root hairs on little seeds. And so next time or sometime in your life you go to plant seeds, I highly recommend setting them out on paper towel first and observing it before you plant them. And once they're in the dirt, you can't see it, right? Okay, but then the other part of the standard that I thought was beautiful that I underlined over here, if we go back, where it says this thermal energy, what we're talking about here is when we talk about metabolism in organisms. So we as mammals are endotherms, and endotherms maintain a constant body temperature. So where do we get that temperature? Where do we get that heat? So when our mitochondria breaks down our food or our organic molecules, it generates heat, and that heat is used to maintain our homeostasis. And so endotherms have a greater calorie requirement compared to ectotherms, which would be like your cold-blooded organisms like reptiles. So now we think about our metabolic rates, and our body shape actually has something to do with this. So when you think about a cute little mouse versus an elephant, and you compare their surface area to their volume. So that really small mouse, its volume, even though it's a tiny little mouse, right? But its volume, oh my gosh, Mary, its surface area is very large comparatively speaking, to its volume. So therefore, as that little mouse is going through and burning through its food to generate heat, it has a huge amount of surface area in contact with the environment, meaning that it is losing its heat to the environment, just the same way like when you are outside and the wind comes by and takes your heat with you to help cool you off. Or if it's cold outside on a windy day, you feel even colder because you're losing heat to the environment. So that little tiny mouse is going to have a very high metabolic rate because it needs to constantly generate heat to replace the heat that was lost to the environment. Versus this elephant, its volume is very large. So when you do math and you compare the surface area to the volume, the surface area to volume ratio is less. So it has, think about the heat being generated in like the core of the elephant. That heat is in indirect contact with the surface and that's outside. So therefore, the elephant hasn't evolved as high of a metabolic rate because it's not constantly losing heat to the environment at the same rate as that little tiny mouse. So anyway, the body size and the surface area to volume ratios matter beyond just cells. It also matters when it comes to thermoregulation. Okay, and so other key points about surface area to volume ratios. You have smaller cells, so now I'm just reading this basically. So smaller cells typically have a higher surface area to volume ratio. So if you were to measure and do math on each of these like cubes, and you measured their, I can't draw, sorry, measured their surface area compared to their volume, it would be more efficient at exchanging materials with the environment. And you can actually check out my surface area to volume ratio video on YouTube. Within that video, I have a time lapse of a lab that does a pretty good job visualizing this. As cells increase in volume, their relative surface area also decreases. But while they're getting bigger, they have a higher demand for resources. So there's a trade-off between how large a cell could actually get. Now, some complex structures, though, are necessary to adequately exchange materials with the environment. So when you think about like the folding of like your intestines, you have like a lot of folds lining your intestines to increase surface area for diffusion. If you think about dendrites, that increases all the surface area of that neuron. You have your the folds in the inner mitochondrial membrane increase the surface area. So really when it comes down to it, if you ever come across a question about increasing surface area or folds, like those go hand in hand. And then part D here is talking about the organisms, which hopefully I did an okay job explaining. Okay, so organisms have evolved highly efficient strategies to obtain nutrients and eliminate wastes. So we're going to look through the next couple slides, different ways that organisms do this, some examples. And then cells and organisms use specialized exchange surfaces. So exchange surfaces could be like for gas exchange, to obtain and release molecules from or into the surrounding environment. So we're going to talk about the kidneys here real fast. So I think this is fascinating to talk about different types of kidneys, because that is how, like mammals, for example, can get rid of our nitrogen waste. So we use urea as mammals, and let me just, oh, I guess I made pictures later, but we use urea and we pee it out. So depending on where that mammal lives, what type of environment, their kidneys have actually evolved to be different depending on their access to water. So this area is called the loop of Henle, and this is one of the areas where we can conserve water in the formation of our urine. So a beaver who lives in freshwater has unlimited access to water. Its loop of Henle has not evolved to be very long, because it is not really at risk for dehydrating. So therefore, by natural selection, a longer loop of Henle was never favored. Where if you compare it to a kangaroo rat that lives in a desert and water availability is definitely a limiting factor, kangaroo rats over the years with longer loops of Henle were selected for. They're more efficient at conserving water. And so this is just an example showing, because if we go back to our standard, it says have evolved highly efficient strategies to obtain nutrients and eliminate wastes. So my little discussion on the loop of Henle is just about eliminating wastes and how it has evolved different in different species. But when we talk about obtaining nutrients and eliminating waste, this can also mean our digestive systems or our respiratory system. And so when we talk about our digestive systems, you can compare herbivores to carnivores, and you can see how like an herbivore that's eating mainly like plants covered in cellulose, that cellulose, we can't as mammals digest it. So as bacteria in that large intestine help access the vitamins in that in those plants, that long intestines helps to give time for diffusion for nutrients. And then the carnivore, though eating meat, if your intestines were super long, like in these herbivores, that meat would sit and kind of like rot in those intestines. And so therefore, they've evolved to be a little bit shorter to have the meat transfer through pretty quickly through the digestive systems. And then obtaining nutrients, we have different types of teeth and fangs. Flat teeth for herbivores have evolved depending on their food. If you look at little sponges in the ocean, they obtain nutrients and eliminate wastes by diffusion. Jellyfish or cnidarians do it. They have these gastrovascular cavities, and basically they have some digestive enzymes lining their cells, and they do endo and exocytosis. Earlier I talked about our kidneys with our different types of, we use urea as our nitrogen waste, but oops, maybe if I put this here, we have different types of nitrogen waste that has evolved in different organisms on Earth. So you have ammonia in aquatic organisms, we have urea in mammals, and then the least toxic of them all is uric acid that you find in birds and reptiles, as well as insects and snails and stuff. Okay, so I think we will, well, I guess we'll keep going. Well, maybe we'll stop here and start a second video so people can like watch this in chunks rather than one long video. Okay.